Welcome everyone to week number 6 in Biology 120 Lab. We've got more DNA going on this week. We're going to be talking about genetics. So last week you learned about DNA. You isolated your own cheek cell DNA, which was really cool. Should have looked kind of globby, mucousy, snot, kind of like. That's what DNA looks like when you isolate it from cells. You also did a worksheet after watching an animation on PCR, which is the polymerase chain reaction, and it's just a technique where you can amplify or make lots of copies of certain regions of DNA, and it's whatever you're interested in. So with COVID, you're amplifying any viral RNA that is present to make DNA copies so that you can detect whether it's there or not. But there are lots of other applications for it. So many applications in the world now. For this week, you're going to be learning about genetics. So how do genes work? Genes are little segments of DNA that give you a particular trait, no matter what kind of organism you are, whether you're a bacterium or a banana or a person or a mushroom or whatever you are. Your genes determine what you are like. And with this, especially in the organisms that do sexual reproduction, you get a mixing of traits from the parents. So the gametes from the parents combine to make offspring. What is a gamete? A gamete is either an egg or a sperm. And you have half the amount of DNA in an egg or a sperm, half the number of chromosomes, and therefore only one of each trait in a gamete. And then when those gametes fuse, an egg and a sperm fuse, then you have the right number of traits and the right number of chromosomes in your offspring. Well, how do you determine what are all the possible combinations of offspring that you can have? We do this in an organized way with Punnett squares, and I will be making some videos this week. I have videos, but they're not of the right traits. But I'll be making some videos this week of Punnett squares that show you how to put together the different versions of traits, how you write down the letters, how you put the letters together in order to figure out what kind of offspring you can have. A monohybrid cross. Hybrid means you have two different versions of a trait. So a monohybrid cross is where you take one single trait of some type and you have two different versions of it. And then you're going to cross two parents that have the two different versions. So my example here, when you put letters to these traits and versions of the traits, you have big T, little t in one parent and big T, little t in the other parent. And when you put those together, what are all the different offspring that you can produce? Well, if you're just looking at that, you can figure out that you can put a big T with a big T with a little t with a big T, a little t with a little t. But with a Punnett square, this is very organized so that you don't get mixed up. A dihybrid cross is basically the same thing, except that you're looking at two different, completely separate traits. So in our experiment with our seeds from the fast plants that we will germinate, we're going to be looking at height of the plant. So are they tall or are they short, also called dwarf? And what's the stem color? Is the stem color dark, or is it dark like purple, or light like white, yellow, green? Okay, dark or light. Totally different traits. But if you have a dihybrid cross, you're going to have the two different versions of two different traits. And how do you put all of those together? And what are the possible offspring that you'll get? And so I'll be doing videos this week of Punnett squares and how to do Punnett squares if you need that. Some of you probably remember how to do this and some of you won't. So I'll put those together and you'll have that as an extra resource. Germinating seeds. I hope that you looked at my video from last week because you know that You needed to start your germinating seeds yesterday. So, or the day before that, or the day before that, because it's going to take you at least 7 days to grow all of your seeds to the point where you can determine what their traits are. So if you're seeing this right now and you haven't started germinating your seeds, start immediately and get those going. All right, watch my Punnett Square videos if you need those when I get them up in the announcements section of the course. I don't have them ready because I have to make new ones this time. And I already explained the 2 traits that we're looking for in our fast plant seeds. It takes a good 4 days for you to see, especially with height, for you to see the traits in these different plants that you're germinating. So that's why you have to get started. And you have 2 batches of seeds to grow. So height and stem color, that's what you're looking for. All right. And then you also have questions to answer from the biology project worksheet. Now, the instructions say to work through all the examples that they have on that website. Oh my goodness. I don't think that I would. You can go to the website and look at those examples when you answer a question. That would be what I would do. I would look at the question and I figure out what information I needed, and then I would go to the examples and work them. Okay? If you write out your answers, which you can do, make sure that I can tell the difference between upper and lowercase letters. And if I can't tell the difference because of your handwriting, then it's wrong, because, you know, that's the whole point of upper and lowercase letters. When you type it, I can almost always tell what they are. But if you write it out by hand, which you can do, you can make your Punnett squares that way. Just make sure I can tell the difference. Draw circles around your gametes. You'll see why I do this when I make the Punnett square videos this week. So there's no question about which ones are the gametes and which ones are the offspring. So what? Remember the difference. A gamete is an egg or a sperm. That is not an individual. That is not a person. That is not a mushroom. That is not a banana tree plant. No, it's none of that. So draw circles around your gametes so I know that's what they are. And space out your answers. The worksheet does not give you enough room to type the answers in, so space them out so that I can read everything, okay? And to give you enough room to answer everything. So those are some hints. And with that being said, I hope that you have a great week. Let me know if you need anything. This is a sort of difficult week because you've got those Punnett squares. And if you are not familiar with them, if you don't remember what in the heck they are, you may have a rough time this week. So let me know if you need anything. m.sigmon@snhu.edu. I'm here to help you. Just let me know. Have a great week.